All right, hi again. Welcome to Pandemic Procrastination Project Part 2. I should be finishing writing a book, and instead I was inspired by some conversations to tackle this today. So what's on your screen here is the process of a clinical trial. This is how a drug goes through development and comes out to market. And this is where a lot of our heads get stuck in studying research. This is really drug trials, and so we're going to walk through it with a drug example. I'm not a drug person. Then we're going to look at what it means when we start to look at wellness interventions, behavioral interventions. So from left to right, we go from preclinical research to phase 1, 2, 3, and 4 research. And each of those phases accomplishes something different. And if you don't like my schematic, go to the Food and Drug Administration, FDA's website. They have another nice schematic. It's much prettier. They have a bigger art department. I just have the cat here. So preclinical research is lab research that's done just in a lab in little dishes and tubes and that sort of thing. But preclinical research is also research on animals. And what that shows is whether there's any potential for the treatment at all. So if you give a drug to all your lab rats and they die, you're probably not going to be giving that to humans. But if it shows promise, then we get into phase 1 research. This is also called T1 research. It's when the first time that new drugs are given to humans. This tests safety. Did they live or did they die? Which of course is not the goal of research. The goal is to make sure that a person can either survive a treatment or at least tolerate it. So these are single group studies, case series studies, randomized controlled trials with placebo. Placebos are important here at some phase in phase 1 because you want to see if the effects are due to chance or something else. But these are generally small group designs. And assuming that the drug is safe, then it moves next to phase 2, whether it's effective at all. So phase 1 studies are done on healthy people who don't have the sign or symptom. Phase 2, sometimes some patients who have the sign or symptom are snuck in there depending on what the treatment is and how dangerous it is. When you get to phase 2, you get to healthy people and then patients who have the condition. This tests the effects of the drug. So I'm going to go ahead and talk about hypertension because that just suits itself so nicely to this. So let's say I have a drug for hypertension that I'm developing and it works well in the lab. So I try it on my guinea pigs in the lab and it works pretty well for them. And I'm going through the same processes, by the way, safety, efficacy, and comparative efficacy on my little guinea pigs. But when I get to humans, I start with safety. So I give my hypertension drug to a single group just to see how well they do. And they're healthy, so their blood pressure drops a little bit, but it's not really dangerous, and that's fine for them. Then I can actually test it compared to a placebo just to make sure that the effects weren't due to chance. I might also, depending on how unique my drug is, have a case series where I start to match people to other people who are like them, just to, again, analyze my data so that my effects of treatment are not due to chance. So it's very, very mathy, research is, and we look for statistical significance. And even when I'm developing the drug, even if I don't get significant reduction in blood pressure, if blood pressure levels off enough for a good length of time, that's still okay. What you don't want to see is patients getting worse in this scenario. So then I move on to phase 2 studies. So if it's safe, I then look at the effects of the treatment and say, all right, now we're going to go ahead and deliver this treatment to people with hypertension. Single group, how did they do? Oh my goodness, they've done really well. Then here we are, randomized controlled trial with a placebo. Again, so I've gone from healthy people to people who have my symptom. Case series might be thrown in here too. I've got a larger sample here. And then at some point we pause and we, the lab, review the research that we've done. And if other people are developing similar drugs using similar methods, as these studies get published in peer-reviewed journals, someone can review the research and decide whether, you as a clinician, let's say, have revealed the drug to be safe and effective, so on and so forth. So the review of research is gathering up a bunch of studies and analyzing them. Tutorials elsewhere for that. Depending on what happens, that might lead us back to modify our treatment. It might lead us all the way back to the lab, back to development. So when we say there's more research needed, you need to point out in the research where it needs to be specifically. But assuming my drug is safe and effective, then I get to phase 3. Now I'm going to compare my drug to other drugs. So these designs from here on, remember we're still going to the right, from here on we don't need a placebo. We don't need control groups. If I had a control group here where I've got a whole bunch of patients who have hypertension and I give them a placebo, what could happen to the patients and the subjects? They could die. So when there is already an available treatment that is known to produce an effect, this is where research ethics pick up. And we want to compare our drug to other available drugs. So it's kind of hard sometimes to get your hands on the other drugs, and this is where research gets complicated. And oftentimes things at this stage get pushed a little bit because I got to go buy my competitors' drugs and use them in my research. But quasi-experimental designs with randomization or not may appear here. You may see large cohort studies. You may see case series. Now you're confused because if you're a grad student trying to do a systematic review, if I don't have a placebo or a randomized controlled trial, how do I find my studies to analyze in my review? And this is where organizations like PCORI pick up to say, we've got to have better methods of reviewing research, because how do you cluster the studies when they don't look like each other? And then it gets even weirder because someone can review these comparative efficacy trials and say either we need more research on my drug compared to even other drugs, or we need to go back and further examine research on whether this effect was due to chance or not. So at some point research starts to go backwards, but it's logical in drug development. It doesn't just go back to the beginning just for yucks. We move on from comparative efficacy, assuming we review the research. Yay, our drug is great, it works really well. Then phase 4 studies are long-term efficacy, more diverse samples, more diverse population, patients who are at risk, let's say. Even different types of uses of the drug. So post-marketing surveillance is used when patients go out and they're given the drug, and they're not specifically enrolled in a study, but they're prescribed a drug, and their healthcare professionals gather data on them at their regular visits to make sure A, they're taking the drug, and B, it's having the same effect. And at some point those data can be culled and analyzed as a group. So what happens at phase 4 and beyond is there's an awful lot of biomedical informatics that get pulled in here, where patients are not consenting necessarily to be in experiments, but their long-term effects of them using a drug are monitored, so that if some weird thing or a danger pops up, that's where it can be caught. So then we have at phase 3 and phase 4, when we're looking at whether it's effective compared to other things, whether they're long-term effects, again, reviews of research. So reviews of research at this stage, if they're going to point back to more research, sometimes that more research is, let's compare our drug to even other drugs or other treatments. But also what happens with reviews of research is that takes us into clinical practice guidelines, how best our drug can be used. So all research isn't necessarily navel-gazing leading to more research. Some research is analyzed to give clinical practice guidelines. Good recent example of that is look at the American College of Physicians recommendations, clinical practice guidelines for non-pharmacologic interventions for non-radicular back pain. It's very easy to see how the review fits the guidelines and how the review is done every 10 years. So then the question is, well, what happens if it's old research? Systematic reviews follow a process. They have a time point where the very first review that's done on a topic, whether you're looking at all of your phase 2 studies or phase 3, the first review considers all available research. Then the next review picks up where that one left off. So now I'm going to pop into an example relevant to massage therapy, just to give you an idea of what happens that's weird in wellness research. So there actually are studies at phase 1, phase 2, and phase 3. There are studies in animals. And this particular example is massage for cancer patients. So when we start with phase 1 studies need to explore whether massage is safe for cancer patients. And here's just 3 citations of peer-reviewed articles that explored that to some level. So what's really odd is that cancer patients are getting massage already, so hasn't this question been answered? Well, yeah. So we move then into phase 2 studies. Phase 2 studies, we're going to focus on well-being here. Does massage improve well-being? Well, there's a couple of studies that investigated that. And in each of these cases, they were looking at whether there was any effect at all. Massage versus nothing. So, okay, you have to have a control group at this phase because this is what you're told. You really don't. You can have a single group design. But the flaw in phase 2 studies in massage is, number one, there's no placebo. You know if you got a massage or not. Number two, oftentimes if patients are recruited from a control group, they decline or they drop out. So there's a methodological flaw here in massage research that has nothing to do with the effects of massage, but it affects the interpretation of the research. So we have enough evidence, and these are also small studies because massage studies are hideously expensive to run. So then we get to this problem of, well, it's not statistically significant. Well, guess what? Cancer patients' well-being is not expected to get better during treatment. Sadly, they get worse over time until they get better. Cancer-related symptoms, cancer treatment-related symptoms. So what happens? Well, if they don't have statistically significant improvements, that means they didn't get less worse. Okay, think about that for a second. So if they're kind of staying at the same level or not quite as bad, that's a really good thing. And that needs to be reported. But unfortunately, it's not reported because people look at this and say, oh, it wasn't significant. So it's really important to educate yourself about the math. But we move on, and there are also phase 3 studies, comparative efficacy. Is massage better than or equal to or worse than other treatments? Can we give patients a choice in well-being treatments? What if you were a cancer patient and you walked in and they said, okay, so during your infusion today, you can get a massage, or you can do art therapy, or you can do something else. You're making that choice, but they should be analyzing the results of that intervention to figure out what might be, where do they put their program dollars in the center? Do they hire a massage therapist? Do they not? So comparative efficacy, there's a paucity of studies here. Now, it's probably tiny on your screen, but look at the dates. There are a couple studies from 2008, 2007. But then if you look back, remember I mentioned before that clinical trials process always moves to the right? But if you look back at the left, you see studies from 2011. If you look back at the left, you see some from 2008. So massage research has an embedded problem. And the embedded problem is that somebody keeps trying to jump into this process at kind of the wrong, or not wrong, but an illogical point. And the illogical point is, oh, we have to always compare massage to a control group. But if we know that it's effective already, then we really need to be moving all the way to phase 4, of which there's hardly, nothing. And this is what aspects of massage treatment result in differences in well-being outcomes. So I'm really passionate about research here. And now this was a massage example, but you can go back through and insert yoga. Is yoga safe for cancer patients? Yes. Does it improve well-being? Yes. Is yoga better to or equal than other treatments? We can go back in and say, all right, let's look at health coaching for patients with diabetes. Is health coaching, is it safe? Well, of course it is. Does it improve, in this case, diabetes? Does it improve, let's say, blood glucose measures, or does it improve adherence to or A1C, or adherence to diet or exercise, whatever the outcome is? And then is health coaching better than or equal to other treatments? So what are the other treatments offered to the patient? So this is what I want you to take away when you're looking at your research question. If you're trying to say you have to always have a control group, then a lot of research, especially massage, yoga, Pilates, exercise modalities, all of those, sorry, that's the cat on the keyboard, all of those are going to affect what we know about these modalities in practice. So hope this was informative, and happy researching.